Hello. The video you're about to watch is part of the Age Presents video series. This series is produced by the American Aging Association to provide an introduction to foundational and advanced topics in geroscience research. As you proceed through these videos, we encourage you to think about the following questions. 1. What relationships between aging and disease are described in the video? 2. What anti-aging interventions are proposed? And 3, what model systems were used in the research and why? We would like to thank our sponsors, the University of Washington Healthy Aging and Longevity Research Institute, the Jackson Laboratory Center for Aging Research, and the Nathan Schock Centers of Excellence at both of these institutions. If you are not yet a member, we encourage you to visit our website and become a member of AGE. AGE members receive free access to our Society Journal Geroscience, reduced membership fees for our annual scientific meetings, opportunities to interact with leaders in the field, all of the lecture materials and slides for this lecture series, and much more. In addition, we have a highly active trainee chapter, which provides numerous opportunities for networking and career development. On behalf of AGE, thank you for watching and enjoy the lecture.
Welcome to this lecture on proteostasis and aging with a special focus on the cellular recycle process of autophagy. My name is Malene Hansen, and I am a professor at a nonprofit research institute in San Diego where we especially focus on autophagy. So today I'm excited to tell you a little bit about, like I said, proteostasis and aging and autophagy, and I just highlighted here a few sort of learning outcomes from this lecture. So I'll tell you about organismal aging as a process that's characterized by molecular damage. I'll tell you about this proteostasis network that relates to homeostasis of proteins in our cell and how it can do that by repair and degradation mechanisms. And like I said, I will focus especially on autophagy as an important cellular degradation process that works in this network. And I will also at the same time give you some highlights on especially how C. elegans, a small nematode, has been successful in gaining insights into aging and proteostasis. So here's a brief outline of how the order of the sections will be. I'll introduce aging as a cellular molecular process. I'll tell you about how C. elegans works as a model organism in aging. And then I'll speak about these different components to the proteostasis network, and especially highlight autophagy with a brief story of some of our recent findings in the lab with a bit of overview at the end. All right, so aging is something we're all too familiar with, myself included. Here's a picture of me 30 years ago now, actually as in my first position as a research technician at a company in Denmark, and now as a lab head here in San Diego. Another too familiar observation is this link between aging and then age-related diseases. So aging is the most common risk factor for many diseases, of many of you would recognize, including a neurodegenerative disease, Alzheimer's, which you can see here once you hit your 50s, the risk of getting this disease gets tremendously increased. However, what's less known is how does autophagy happen at the molecular level? What are the changes to our cells that takes place over time? And that's really what I will talk to you about today, just noting that we and many other research groups are of course doing this with the aim of getting such information to help not only understand human aging better, but also help fight some of these many age-related diseases. All right, so here's a cell, and as you will know, all of our tissues in our body are composed of cells that has different components to it and compartments, as I will also talk about, including the nucleus where chromosomes are localized, on which DNA is situated. And to that end, you will probably also know that the DNA codes for different genes. So for instance, gene X here, each of them produces a protein that's going to fold right. We'll talk a lot more about that, and each of those proteins have a function. So an analogy would be the DNA keeps the information, right, that codes for the tools that's being made, and they each have functions within our body. Now, over time, though, as we all recognize, there can be damage to the cell. We are exposed to different physical, chemical, biological agents that would change the cell over time. And so I'll just highlight with a few visuals of some of the hallmarks that at least right now we're considering that are contributing to these changes over age within our cells. So one is DNA damage. I already mentioned that. UV exposure, for instance, can cause changes to our DNA. Other problems in the nucleus relates to telomere shortening. So the ends of the chromosome has specific issues in terms of being maintained. This can overall alter gene activity, which again is important for the functions of the cells indeed. Protein damage can happen. This will be the key point to a lot of the rest of the talk in terms of protein homeostasis. And then altered signaling within the cell that also ensures how it functions can be critical, along with changes to other compartments like mitochondria. These are the energy factories of the cell that can dysfunction and cause both problems inside, but also aberrant functions to proteins and other signals that actually leave the cells, and you can imagine can cause problems beyond your own cell at an organismal level, as we know aging to be. Okay, so that's sort of the bad news. The good news is that the cell, over many years of time and evolution, have also developed processes and have genes that can help repair and sort of counteract this damage. And that's what I'm going to tell you a lot more about today. So what are the genes and repair processes that can play roles in aging? So over a number of years, 3 decades, there's really been an extensive search for these so-called longevity genes by using simple model organisms. And one of them being C. elegans, a specific and small nematode that our lab also uses along with many other labs. It's a tiny animal that lives in the soil. Here's a picture of my hair, and you can see how small they are, about 1 millimeter long. But they have been tremendously important as a multicellular, also transparent animal with many conserved molecular mechanisms that include other events to the cells like apoptosis or cell death, how synaptic transmission of neurons happen, and also aging, as we will talk more about. Another feature is that they're very experimentally tractable for genetics, biochemistry, and also RNAi, as I will talk later about. This is a way to basically inactivate genes by means of an RNA-related mechanism, and we can do that by feeding the worms. They eat normally bacteria. We can feed them full of RNA, and in that way affect gene expression, as you will see. They were also the first multicellular organism to get their full genome sequence, which again facilitated genome-wide RNA libraries. Again, that can be helpful in asking what are the functions of different genes when we turn them down and ask for their function. But really, and I should also just highlight, they have been tremendously prominent in also being involved in 3 Nobel Prizes in just 6 years, including the full cell lineage for the worm itself, and also the RNAi mechanisms, as I just mentioned it, as well as GFP, the fluorescent protein GFP that has been used to provide a lot of information in not only worms, but in many other organisms. But it was actually first done in worms. However, for our research and those of others for the purpose of this topic is really that these worms ages really fast, only in a couple of weeks. And you can see that it's accompanied with a number of changes that even to a non-trained person, you can see this looks like an old worm. They seem to look more wrinkled even at the microscopic level. Their muscles tend to fall apart in many ways like ours during sarcopenia, and they also suffer more infections and reduce mobility over time. Again, at least in some ways, paralleling the pathologies that are observed in human aging. Now, one of the key observations to the field more than almost 30 years ago, was this idea that single gene mutations actually could change the lifespan of the animal. So what I'm showing you here is a lifespan curve, the fraction of worms alive over time. And again, key to this organism is that it all happens over a number of days. So a wild type animal will start to die. Some will die, be unlucky, die early, and they will create this lifespan curve that is not much different than a human population, except that the speed by which this takes place. But these animals that were identified in the and characterized in the Kenyon lab was to show that a gene mutation in a gene called DAF2, you can see extended lifespan. The animals started dying later and at a slower pace. Gary Rufkin's lab cloned this gene to show that it was an autolog of the insulin IGF-1 receptor, and really it was the beginning of looking at this insulin IGF-1 pathway as a pathway to longevity. And there's a number of genes already identified to this. I'll just highlight one. The transcription factor called DAF16 or FOXO is completely required, as you can see here, for the long lifespan. So if you carry the DAF2 mutant but don't have the transcription factor, you can see you have no benefits. So this is a very simplified model or diagram of this pathway, which in work following this was shown to basically have similar effects on longevity in other organisms, depending on tissue and intervention. Perhaps also in humans, where longer humans called centenarians that live beyond 100, there are also examples of having mutations in some of these genes, including the FOXO transcription factor, seems to be correlating, not in a necessarily causative way, but correlating with the long lifespan of these humans. So that's really an interesting observation and the beginning of the genetics of aging in many ways, where a lot of work has elucidated not only reduced insulin IGF-1 signaling, but also other genes and processes, especially highlight dietary restriction, which was started to be looked at even in the 30s, with a number of other mechanisms also added to it. Reduced mTOR signaling. mTOR is a sensor of nutrients to the cell and greatly linked to dietary levels, whereas other mechanisms like reduced mitochondrial respiration, another important process for the cell, as well as removal of the germline, are just examples of what we call conserved longevity paradigms, meaning they all, in a number of different organisms, lead to longer longevity. And the reason I put these up was just to highlight that many groups, including our own, are interested in understanding how these processes extend lifespan, and also to what extent they overlap. And one of the overlapping pathways, which is the purpose and the focus of today, is to talk about how they can improve protein homeostasis using the phrase that is in the title, namely proteostasis. And this has been done to a great part in C. elegans, but also in other organisms, as I will just briefly highlight. So here's a brief summary so far. Aging is associated with increased risk of a number of diseases. During aging, there are many of these cellular functions that declines, and that causes a problem with the homeostasis overall. A lot of studies have been done in the short-lived nematode C. elegans, and through this work and the work of many others, including other systems, is to show that there's a number of conserved longevity paradigms that exist and that we can study to learn more about the pathways of aging, including those important for proteostasis. So switching now to proteostasis, which is key for how the cell can maintain its proteins. And so we all know protein is made by synthesis that would create a normal protein. But again, as I highlighted earlier, damage can create toxic protein to these molecules. However, the good news is they can either be repaired or degraded to alleviate this problem to the cell. And I'll go into this in a little more detail now with some of the more detailed references, and I am providing a reference list to this end for those of you who are interested. But as I highlighted before, again, in the cytosol, the proteins have to be made and translated into these nascent polypeptides. A high number of chaperones within the cell are now important for folding these proteins into their native proteins, or if for whatever reason they have unfolded unproductively, there are chaperones that can help refold them. And so this is, as you can imagine, very important for maintaining a functional set of proteins to the cell. Now, this can not always or does not always happen right, and the cell, like I said, has mechanisms to then try and abort and kind of remove those problematic molecules. One prominent system is the ubiquitin proteasome system, UPS. A high number of proteins are involved with this, and basically involves a way of tagging these proteins with so-called ubiquitin small tags that would target these proteins for degradation by a number of means. And I'll show you in the next slide how that kind of seems to look like on a molecular level. Proteins can also aggregate in a more prominent way and kind of either make, these kind of amorphous aggregates or oligomers or even fibrils that are even more complex structures of these proteins. And while there's a discussion in the field which of these are perhaps the most toxic, you can see they may not always be productive for the cell, and in that case, therefore, there are also mechanisms to help alleviate or degrade and remove those. It's specifically by this pathway called autophagy or macroautophagy. And I'll tell you a little bit more about how that happens in this slide. So this is kind of a representation of the same. In the cytosol, proteins are made by protein synthesis. They can misfold, in which case they can be helped by chaperones that again brings into this so-called ubiquitin proteasome system that looks like a little, you can see a little trash bin with a lid on that will fit in these proteins and help them degrade to remove them from the cell. There's a number of other mechanisms, like I mentioned, combined called autophagy, of which there's a couple of different flavors. So this one highlighted is a mechanism by which chaperones can actually direct misfolded proteins directly to the downstream organelle here. That's the lysosome, and that's a molecule or vesicle that contains basically acidic hydrolases that can help degrade these proteins. The other way that can basically take care of much bigger aggregates is macroautophagy, and I'll talk to you a lot more about that, by which double membrane structures are formed that basically sequesters the so-called cargo that then fuses with the lysosome for degradation. Again, both mechanisms by which you can remove problematic proteins. Another way, as you can imagine, and this is a very current topic of the field, is if you can't deal with it sort of internally, the cell also can basically try and send it outside, right, to sort of dump the trash outside of the cell, for instance, with small vesicle called exosomes. And you can imagine that would be good for the cell here, but as an organism, you now have to deal with, obviously, these exited aggregated proteins in other ways. And like I said, this is a hot topic, and I won't talk much more about it today, but it's really an important thing to think about in terms of how cells communicate and organize also aging from an organismal perspective. Last but not least, there are also organelles within our cells, like mitochondria, or what's called endoplasmatic reticulum. So this is a network of membranes that ensures that proteins that are meant to either be put on a membrane or sent outside as of being excreted can, of course, also misfold. And there are, again, systems within those organelles that can help kind of alleviate that. So ERAD, or ER-associated protein degradation, is one mechanism by which also there's communication via the unfolded protein response to the nucleus in terms of chaperones that help maintain homeostasis in this organelle system. Or newer data ensure or have highlighted that you can also send parts of the ER to the autophagosome for degradation, again ending up in the lysosome. Another prominent example is mitochondria that can be removed by mitophagy, again by means of being tagged for degradation, and it ends up in the lysosome. Again, there are other mechanisms here that are mitochondrial unfolded protein response that again communicate with the nucleus for maintenance of homeostasis. Needless to say, you can imagine that so intricate systems if they don't work right, whether they be in the cytosol like chaperones or the UPS of autophagy or in organelles, you can imagine that any malfunctions, and there is some highlighted here, can cause a number of diseases. And there are plenty of examples, as you can see, for age-related diseases I already highlighted, like Alzheimer's disease or other diseases like Parkinson's disease. They are associated with aggregations and these kind of oligomers and amyloid fibrils, like I already highlighted. The good news, and of course, again, of promise for the future, there are ongoing drug trials to try and target different components of the proteostasis network, both for the chaperone system with heat shock factors and also in autophagy, for instance. And these are collectively known to sort of try and combat these neurodegenerative diseases, which are also often called proteinopathies in general. All right, so here's a brief summary of the proteostasis section. It's a network that can help the cell maintain its homeostasis. It can remove misfolded proteins, try and refold them first of all, and also remove aggregates. There are many basic biological processes that are important: protein synthesis, refolding by these chaperones, and protein degradation, of which you can either remove misfolded protein smaller entities by ubiquitin proteasome system, or via autophagy, which can take care of bigger issues like protein aggregates and even whole organelles, like I mentioned just before. And that's been tightly linked to many age-related diseases, as I mentioned. So back to proteostasis and the long-lived animals, where a lot of the more details have now been looked at, and that's what I'm going to try and highlight in a little more detail with a focus on macroautophagy. So just a little bit of history for macroautophagy, which recently was awarded to Professor Osumi for his work in elucidating the basics of this process. So the process itself was originally coined by Christian Deneuve. Electron microscopy studies have basically showed that there are these presence of double membrane structures that appear to contain parts of the cytoplasm and also organelles in these various states of disintegration. And so Professor Usumi was trying to study these macromolar proteases in Japan in the late 80s, when he wanted to see if he could find links to these enzymes that could perhaps be important for degrading organelles within the yeast vacuole, where the proteases were mutated or blocked. And really the best part was that he could look at these vacuoles basically by light microscopy and basically do genetic screens for genes that were important for mediating this degradative process. And so here's a highlight of that. I'm not going to go over all of the different proteins. He's identified a number of them, but more highlight the process itself with initiation of upstream signaling, including by this mTOR nutrient sensor I mentioned, that will cause isolation membranes, so small pieces of double membrane structures. It's still a debate where they would come from, for instance, from the ER itself, or the plasma membrane, for instance. This membrane will elongate and expand. It's called the phagophore, that eventually will close as an autophagosome that goes on to fuse with the lysosomes I already mentioned. Little sort of Pac-Man enzymes or basically acidic hydrolases would be present in these structures that can fuse and in that way help with the degradation of the cargo inside. I'm just going to highlight one protein, the protein LC3, that via a number of conjugation system is going to be attached to the membrane. It's proteolytically basically processed, but then attached to the membrane of this autophagosome, or in this little cartoon. This is what it highlights in a C. elegans worm, where you can basically look at them as little punctae that highlights these structures of autophagosomes, or the pre-autophagosomal structures. So that's one way. It's also exchangeably called macroautophagy, where again you recruit different components of cargo into these autophagosomes. There are selective forms to this. This is the way to categorize if the cargo is known. So for instance, mitochondria, mitophagy, I already mentioned, are aggregated proteins, which would be macrophagy. There's two other main parts by which the lysosome helps to degrade this. Macroautophagy would be a different kind, where you basically recruit components of cargo directly into the lysosome that invaginate basically in macroautophagy, Or there is a more selective, even more selective form that's chaperone-mediated autophagy, where co-chaperones basically direct here to a specific receptor that helps, again, enter the protein for degradation into the lysosome. Okay, so not surprisingly, autophagy itself, just like I mentioned, the other proteostasis pathways in the network have been linked to a number of diseases, often in quite complex ways, and also directly to aging. And here's just a highlight of some of the links that's been made. Autophagy is required for all the long-lived mutants I already mentioned to you. There are ways to activate autophagy that seem to promote longevity, and autophagy declines during aging and in age-related disorders. And I will go through a few examples of this. So by means of autophagy, we and others have shown that if you inhibit autophagy during adulthood, and of course this is important for not interfering with this process that also have roles during development, there is little to no effect on a normal animal. Again, we do this with RNAi, so you ensure sort of not a full loss, but like a reduced activity. And in that case, for all of the long-lived mutants we have looked at so far, these are the examples I showed you in the diagram, the lifespan is basically lost if there is not a full working autophagy machinery. And this is not only true in worms, this is a highlight of some of the other mechanisms or other model organisms by which inhibition of autophagy or autophagy genes have been shown to be required for this longer lifespan. The flip side of this is, of course, to ask if autophagy induction is sufficient to lifespan extension. There's been a couple of examples where genes important for autophagy has been overexpressed. For instance, the transcription factor TFET, that is a known kind of master regulator of many autophagy genes, which we have shown extend lifespan, and these in part by autophagy induction. And a number of other genes, including in mice, have been overexpressed to show that there are links, perhaps not causative at this level, but that there are links between autophagy genes and aging. All right, so this is sort of the highlight. I wanted to highlight a little more detail how autophagy declines with age and how we think about that as a way to now figure out how it fails during aging. Obviously, again, with the perspective of how we could potentially fix it in the long run. So our lab has, together with a number of other labs, worked and developed a number of reporters, again, linked to this protein ATG8 that is attached to the membrane. It's called LDG1 in worms. And we basically use that along with some other methods that I would highlight right now to show that autophagy seems to decline in wild-type animals. And so it decreases with normal aging, such that in older animals, you don't have much activity left. And so the way we deduced this or proposed this was to use this reporter again and look at a number of these different tissues of the animals, including the intestine, its muscle, its pharynx, which is its feeding apparatus, and also in neurons. And as you can see over time, you can see that the number of these autophagosomes highlighted with this marker, they seem to increase over time as a general notion. You can also appreciate that it doesn't seem to be happening with the same kinetics. Muscle seems to be especially different or change a lot. We don't know the reason for this, but we speculate that that's because of the different functions that autophagy plays in those tissues. But one notion to an assay like this, which is what's called a steady state assay, is that we're just counting basically these autophagosomes by means of these green dots as a means of how many are there. But you will remember that autophagy is a very dynamic process, so you can imagine that you can get more even by forming more or by not being able to turn them over. That would essentially look the same if we just took a snapshot like we do here. So the question is, what did this increase in autophagosome represent, an induction or a block? And so we have basically implemented an assay that's used well in mammalian system also, that basically is the principle of how this isolation membrane forms the autophagosome, fuses with lysosome. And if this running process, you add an inhibitor, for instance, the chemical Bafilomycin A, it will inhibit the acidification of these autolysosomes. You would basically block the process and accumulate everything that would be before, like the equivalent or analogy being like a conveyor belt. If you stop it, you would just have more products on the belt. However, if the process is already stopped for whatever reason, and you now give an inhibitor, nothing or much more should happen. And we do this again. We inject it directly into the animal with a red dye that's bigger than the BAF molecule to see that it had reached the tissues we're interested in looking at. So a former postdoc, Jessica Chan, did this and basically injected animals over time. And as you can see, there is an increase in these dots in all the tissues, but perhaps less so with time. And that's something we were really interested in to understand further, but we took it to mean that autophagy activity is basically running in all tissues. This is consistent with other data, both in C. elegans by Dana Miller's lab and others in systems of, for instance, mice. And then it seems to generally decrease with age. That's at least our hypothesis. And so that's something we're actively working on now to really try and ask how and where in this multi-step process could autophagy decline. And I already pointed out the different steps to the process. Now, I told you that our data is consistent with an accumulation of these kind of structures. So perhaps, the initial steps are fine, and there are some data in other systems to that end. And so our current efforts are really more focused on the later steps, including on the lysosome, where there again is published data from other systems, including from Dan Gosling's lab in yeast, that argues that there could be changes to the lysosome that, of course, again, could cause an overall change to autophagy and perhaps an accumulation of these earlier structures that are kind of half-baked and can't go through the whole process per se. But this is ongoing for sure. I won't show you the rest of the data, but we also looked at these, used the same kind of methods to look at long-lived animals that seem to be able to maintain these processes for longer. And so that's kind of the highlight that I already mentioned to you, that there's better macroautophagy in these long-lived animals, and we actually don't have any exceptions to this at this current point. The question, of course, and that's what our lab is really interested in working on, is how is this regulated? What is it that's being turned over? And where and when in this multicellular organism does this happen? And there's a lot of impacts to this, including knowing about the regulators, right, could help us think of or help develop therapies for many of these autophagy and age-related diseases. Knowing about the cargo would be important to understand more about how the cell can be protected by these mechanisms if they go wrong. And the impact, of course, would be huge in terms of having knowledge about where and when to affect this process would be very important for how we could go to higher organisms eventually to develop interventions towards these diseases. So I'll just end by a small highlight of some of our latest interests, and also the current sort of focus of autophagy field in great part is about so-called selective autophagy. So this idea that there are specific types of cargo that is being turned over by autophagy, perhaps in an age-relevant way. And so we have screens to this end, but also have candidate approaches, which I will share with you today. So this relates to another way of making animals longer in a conserved way. Mild heat shock is something you can give to an animal, a very small heat shock that later will prepare it to be basically more resilient. Think about it like what doesn't kill you will make you stronger. So this has been studied especially in the context of the chaperone system by Rick Morimoto with this classical heat shock response. But a former postdoc to the lab, now a research assistant professor, initially found a few years ago that this also caused an induction of autophagy, and this was important for this hormetic adaptation to give better long lifespan and also improve proteostasis. So one of the genes that was the highest induced of autophagy-related genes was this gene SQST1, which is an ortholog of a gene called P62. That's a known receptor for autophagy. It is a protein that basically bridges this LC3 protein that I mentioned is a protein that attaches to the double membrane. And then on the other side, it binds to ubiquitin, the tag I already mentioned, that ubiquitinated proteins that aggregate gets tagged with for degradation, or alternatively whole organelles, like I mentioned, mitochondria. So this protein also exists in worms, where it pretty much does the same thing. It binds to the ortholog LTG1 on the membrane and recruits aggregated proteins at least during development. So we were interested in looking at how this this selective autophagy receptor could perhaps be important for heat shock-induced autophagy. And so we found basically that heat shock that otherwise we had shown induces autophagy does not happen in animals that doesn't have this receptor. We did this in a number of tissues, the intestine as well as the neuron. And as you again can see, there is no response to giving this heat shock without the receptor. Importantly, also, there was no benefits from this. A hormetic heat shock gives a lifespan extension, as I mentioned. This is the hormetic benefit from this small hormetic heat shock, 1 hour early in adulthood, as you can see, from black to gray. But again, if you're a receptor mutant or you don't have the receptor, you don't enjoy any benefits. This was interesting also because it didn't seem to be the case for many of the other longevity paradigms I already mentioned to you, where we also tested if this receptor was required. So there, those mutants all require these canonical core machinery component of autophagy, but not the selective autophagy receptor. So one idea is that they could require other receptors that we're currently trying to identify, or they function more by more general autophagy mechanisms, something that we're very interested in right now. And then the final phenotype that Caroline Kumsta looked at the time was following up with the interesting observations she had found in her first paper, which was that this hormetic heat shock seemed to reduce the number of aggregates that you could observe using polyQ. So polyQ is associated with Huntington's disease, proteins that starts to clump because of a number of these repeats. This can be recapitulated in worms, for instance, by expressing them in neurons. These are strains from Rick Morimoto's lab, where you can see over time you start to get these aggregates. However, with a heat shock, we could find that this reduces the load of aggregation. Again, not if you're a receptor mutant, though. If anything, you just have more aggregates. So again, the receptor seems to be important for dealing with this aggregation load that we can artificially put on in the C. elegans system to that end. So that's just a recapitulation here. The heat shock induces autophagy, in part increasing this receptor P62 levels, and that helps with these beneficial outputs. That prompted us to ask that perhaps if we overexpress the receptor by itself, this would be sufficient to recapitulate some of those effects. And indeed, that was the case. We could show that overexpressing P62, the receptor, at least at 20 degrees Celsius, you can see a lifespan extension that's dependent on the mutant. Importantly, we tested a mutant we created that didn't have the domain that's important for binding ubiquitin. So remember, I explained how that's how it basically catches its cargo and brings it to the autophagosome for degradation. So that's consistent with an important role in protein degradation. However, it didn't discriminate with whether it brings proteins for degradation to one degradation mechanism, which is autophagy. The other one is the proteasome, which I already mentioned, that also can take care of ubiquitinated proteins. But we did some additional experiments with a collaborator, David Wilches, in CCAT, and he analyzed these animals to show that they didn't actually have high effects on the proteasome activity. So we believe that this relates a lot to inducing specifically autophagy. And consistent with that, we could knock down different genes in the pathway. Again, this is a multi-step process. So, you know, we test the gene in all of these different steps. I'm showing you here the result of the gene LDD1 or LC3, and you can see again, like I showed earlier, without the receptor, here, if you take away autophagy genes, machinery, the overexpressing animals don't have any benefits. So the last bit we did to that end was to test directly what autophagy looked like in those animals. We used, again, the method of looking at these little dots with our receptors and did the flux assay to show that the increase actually that we see from gray to green in these overexpressing long-lived animals could be further increased when we give this autophagy flux BAF-A-inducing agent. And you can see there is induction in both cases, suggesting that there is increased autophagy in these overexpressing animals of interest. These are really huge autophagosomes that form, and we're really interested in analyzing them in more detail. But the really, I think, prominent part to this study was to show and observe for the first time that the receptor seemed to be playing a more instructive role in regulating autophagy, not just simply facilitating bringing the cargo in for degradation, but maybe even having a more active role in inducing autophagy, which there is a few lines of evidence in the field at this point, but the detail is definitely not known yet. So that's just a highlight of what I showed you in worms, that we can induce autophagy by hermetic heat shock. This includes the overexpression of P62 that's sufficient for lifespan. We looked at macrophagy and improved proteostasis to that end. Recent work in parallel, actually, from David Walker's lab in Drosophila for fruit fly showed similar links that also overexpression in that organism could ensure longevity. So perhaps emphasizing this as a potentially conserved mechanism. There's a lot of things still to be learned, and I highlighted already earlier this idea of how cells communicate with each other. In this case, we found specific roles for this receptor in the neurons. And so we're really interested in understanding how these neurons or other tissues signal back and forth between neurons and other major tissues like the muscle or the intestine of the animal. So these are ongoing objectives, not only for the sake of autophagy, but certainly also for the rest of the proteostasis network. So I am going to close here and just leave you with the overall take-home messages of my whole seminar or lecture. I have highlighted how aging is accompanied by a number of loss of cellular processes, including proteostasis. This network is comprised of different molecular pathways that ensure protein synthesis, refolding, and also degradation. And there are many age-related diseases that are linked to failures in this network, in particular these neurodegenerative disorders of proteinopathies. And then I highlighted how especially the short-lived model organism C. elegans have been extensively used to study proteostasis and its regulation with age. And in particular, the long-lived C. elegans have provided really important and useful tools for us to learn more about how one could potentially maintain or boost the proteostasis network in a beneficial manner, including or in specifically for the example I showed you, to induce autophagy potentially in a very selective fashion that we can perhaps target, with the ultimate goal, of course, to also focus our efforts towards healthy aging of humans and disease prevention. So with that, I'm just going to close, highlight the acknowledgement of all the people, at least in my lab, that's been helpful for our studies, as well as all my other colleagues in the field that have really provided a great foundation for how we think about proteostasis and aging. Thank you so much for your attention.